Hello, I am Satish Kulkarni. In this lecture, we will explore about classification of biological databases. The databases are classified into different types for the sake of maintaining quality, improving accessibility for information, and reducing redundancy. There are varied modes of database classification in published literatures. Therefore, it is essential for a learner to identify and search and retrieve the information in the relevant database rather than dwelling upon its hierarchy. The categories of databases for life sciences are sequences, genomic, mutation or polymorphism, protein domain or family, proteomics, 3D structures, metabolism, bibliography, and others. Based on type of data, sequence of biomolecules, biomolecular structures, bibliographic scientific literatures, patent databases, varied types of databases are being created. Their linkages and their names have been narrated in this table. Before we go for the classification pattern, we will look upon the features of biological databases. This includes data heterogeneity, high volume data, data uncertainty, data curation, data integration, data sharing, dynamics for new data. Data heterogeneity is availability of diverse and complex data types. Data types in biological sciences is sequences, graphs, high dimensional data, different shapes, temporal data, patterns, scalar and vector fields, extracted feature data. High volume data mean, in addition to being highly heterogeneous, biological data are voluminous to support comprehensive investigation in various fields and directions. As far as data uncertainty is concerned, biological data have a great deal of uncertainty as they represent biological phenomena that are observed and assumed. Data curation includes biological data collected from various sources across the different structural and functional boundaries. There is always a chance of missing links. To fill this, data is analyzed and curated via automated methods. Data integration includes, after the years of research across the different structural and functional scales, data is collected from laboratories worldwide and integrated together through a database and made available for use. Data sharing is consisting of biological data is shared via databases. The purpose is for scientific communities inspection, for cross verifications, and to prevent repetition and validation of the data. The dynamics for new data includes the new data is continuously generated in the laboratories and sometimes this new data contradicts the old data. So it is necessary to develop the organizational database schemes to incorporate into the new data. The databases are classified using variety of criterias, but the main two criterias on which it works out is type of data or type of information or source of data or source of information. Databases actually are classified into primary and secondary databases, and based on the different criteria like structure of the databases, type of the databases, quality of the databases, and maintainer status. The classification for biological databases can be looked upon based on these different points. The first one is the data type, second one is a maintainer status, third one data access, fourth one data source, fifth one data design, sixth one organism, and the last one is data content. The biological databases are also classified based on the data types into genome databases, sequence databases, structure databases, microarray databases, chemical databases, pathway databases, enzyme databases, disease databases, and literature databases. Based on data type, we have all these, and each individual class we have different examples like nucleotide databases, we have GenBank and EMBL. Protein database, we have SwissProt and PIR. Structure databases, we have PDB, NDB, DALI, MSD, etc. Microarray databases, we have ArrayExpress, etc. Chemical databases, PubChem is there. Pathway databases, KEGG, BioSilico is there. Enzyme databases, ExPASI, Rebase is there. Design databases, OMIM, OMIA is there. Literature databases, PubMed and Scopus is there. These are the different logos of the database nodes present around the world. As far as maintenance status is concerned, we have NCBL and EMBL. These are academic groups of scientists who are maintaining this particular databases. We have a data access. Based on data access, we have publicly available databases available with copyright, browsing only, accessible but not downloadable, academic but not freely available, proprietary, commercial or restricted kind of availability. Based on database design, we have flat file databases, relational databases, object-oriented databases, or exchange or publication technology based databases. Based on data source, they are classified into primary database and secondary database. Primary database consists of original data submission by the researchers, and secondary database is results of analysis of primary databases is included here. Based on organism databases, we have bacterial databases, virus databases and human databases. Based on DNA content, we have protein, DNA, RNA, microRNA databases. We have a protein family databases. Based on common physical properties like membrane bound mitochondrial proteins, we have the databases arranged. Common chemical properties like protease, reductase, etc. they are also there. Sequences of particular genome, species, influenza sequences, plasmodium sequences, these are there. Motif and domain databases are also there. Primary databases, these are containing biomolecular data in its primordial or original form. They are mostly public databases, hence they have got open access. Primary nucleotide sequences contain heterogeneous mixed data including whole genome sequences, gene sequences derived from the genomic DNA, mRNA, sequences of chromosomes, complete partial sequences, annotated or unannotated entries with established or predicted functions. Therefore, identification of the sequence of interest from primary databases involves screening of large number of entries. Different examples are there which are quoted here. Secondary databases contain information which is derived from analysis of primary data and are therefore considered to contain more relevant and useful information structured to specific requirement. These are manually created and automatically generated. Representative examples are given like ProCite, prints, blocks, etc. Composite databases represent the amalgamation of several primary databases. They are easy to use. The composite databases allows users to access all the relevant information from the single source rather than connect the multiple resources. One of the best example of composite databases is NCBI. And the fourth one is integrated databases. These are containing the data that has been collated from different but related organisms. Such data is very useful for comparative genomics studies and provide better insight into the evolutionary relationship and synteny between the genomes of different organisms. Such studies are useful in evolutionary studies. Examples are quoted here. These are some set of databases which are organ specific databases because they are being created for some specific purposes applicable to different kinds of organisms. In this table, we have the databases and their URL, and then we have organisms for which they have been designed. Then we have organ specific databases like on green alga, Chlamydomonas, we have Chlamydomonas Center. Then we have a medicago.org, Barrel Medic. This whole list goes out for organ specific databases. What to look for the databases and where to look for these databases. We can search these databases using varieties of search engines. We can also find out the information regarding the databases in journals. We can look out for the different websites where most of the biological databases are available through the websites, and several other websites are also being designed to give the information and accessibility to the database. One of the very prominent journal wherein the database information is being published, this is Nucleic Acid Research Database issue. Every year this is being published which is printing out the complete list of databases which is being created. These are 10 important bioinformatic databases along with their URL and the type of informations available around the globe. This is the work cited for classification of the databases. Thank you.